All right, welcome everybody to this KBase webinar. Today we're going to hear from Dr. Adam Arkin. He's a senior scientist based at Lawrence Berkeley Lab, as well as the University of California, Berkeley. He's the principal investigator for the DOE Systems Biology Knowledge Base, aka KBase, the system we're talking about today. And he's also a lead on several other projects, including the Ecosystems and Networks Integrated with Genes and Molecular Assemblies project. That's Enigma. So we'll be talking a lot about Enigma data today. So if you want to look up Enigma, it's on the Berkeley Lab website. You can find out more about that project. And he's also a lead of the Center for Utilization of Biological Engineering in Space, which is a very exciting program about microbes in space. And of course, like I said, he's the PI of KBase. He's a real leader of the project and a real source of the vision that kind of underlays all of the technology and features that we have on systems. So I'm going to turn it over to you, Adam, if you want to share your screen and let you take it away.
Hi, thanks, Ben. Obviously, the vision comes from the entire team, not just from me, and especially my co-PIs here, Chris Henry and Bob Cottingham, and our leadership team, Parnder Dahal, Shane Cannon, Alicia Witch Charlson, and others. So I also want to thank you for sharing, for West Coast people for sharing the lens of me, and for the people who are East Coast for sharing your afternoon doldrums. We are going to take a bit of a whirlwind through a complicated scientific workflow that we're still building out in the KBase. Our goal is really to begin to build predictive mechanistic understanding of how organisms operate in open environments or complex environments, even if it's not open environments. And we're focusing right now on problems that are about mechanistic ecology, that is how microbes find themselves in the world, how they're dispersed to different locations, how they grow there, and how they interact with other microbes. And we're trying to develop a workflow that would enable you to begin to integrate lots of different data sources together, both your own and other people's, to begin to better understand what the keystone objects are that drive the activity and function of a given community in a given location. That's really what we want to do. And so we've been designing KBase to enable teams of scientists to collaboratively work together, to bring their data together, and to organize them and share them with each other so you can build these sorts of workflows. So I'm going to take you through a bit of that story using a program that I am very intimate with, which is the Enigma program. As I said, KBase is a fairly diverse team, and we have a number of scientific area experts. We have people who are developers, people who are outreach coordinators and educators, and it's extremely interactive and very integrated and a real amazing group to work with. We are also very much, we're actually next door neighbors, integrated with the Joint Genome Institute. We're distinct programs, but we share a space, that beautiful IGB building, if you're at Berkeley. Obviously, we have people at Oak Ridge National Lab, at Brookhaven, at Arkin National Lab, and so we are dispersed, but we have very strong alliances with the JGI and the National Microbial Data Collaborative, which is another fantastic resource for people involved in this line of research. Now, as I said, what our specific goal is to empower scientists and teams to collaboratively drive discovery, prediction, and control and design of functioning plants, microbes, and communities, and to do so as an open way as possible, and to be able to, as quickly as possible, propagate their results to others in an effective way. That's what we're trying to do. So a lot of the design that you're going to see here can seem complicated at first, but it's in service of you having full control of your data, but being able to move it out to your immediate collaborators into the world as quickly as possible and integrate with their results. That's our goal. So we're going to talk a little bit about this idea that we want to make it very easy for people to go from data that's out in the world, say the field, it could be out in a terrestrial location or a marine location or in, in the human gut, and translate that into, and integrate it with laboratory information that gives us a deep mechanistic understanding of the microbes and how they operate, and then move that back again to predict what's going out in the world. I'm going to try to show an example of what I mean by this by using the Enigma data. Now, for those of you who are not familiar with Enigma, Enigma is a large SFA, a centered focus area for DOE. It's been running in one form or the other since 2002, in its current form since 2011. And our goal is the mechanistic and predictive microbial ecology of metal and nitrate contaminated subsurface, the shallow subsurface. This map that I'm showing you over here is a map just of the United States, where the sort of orangey color is uranium and the bluey color is nitrate. And where you're seeing the very deep colors, that is where we have a problem, where they're above healthy levels. And so obviously, because of where I live, I'm interested in this one over here, which is where this is the California agricultural corridor, where because we've over fertilized, there's a high degree of nitrate in both the sediment and the soil and the groundwater, which is actually leading to poisoning of the people who work the fields, for example. And when you do add nitrate to sediment and soil, it's acidic, it forms nitric acid more or less. And when it does that, it dissolves the minerals, and among those minerals it dissolves is uranium, the United States being a uranium-rich location. That's pretty bad, of course. You can see all the little areas where it's pretty bad for uranium or for nitrate. This big thing over here is the Luala Aquifer. It's the major water catchment that serves our breadbasket. And for the first time in the last last decade or so, Kansas farmers are pulling up uranium in their corn. We've overpumped this well by a huge amount. It's not going to be replenishable. It has become extremely acidic, and it has become extremely nitrate contaminated. And we have to vastly change our water resource utilization if we are going to, see if we use this resource effectively. We are not in any one of those locations. We're actually in this little dot over here. That little dot over there is Oak Ridge, Tennessee. This is the so-called Y12 site. So the Y12 site is right next to Oak Ridge National Lab. It's a classified location where the Manhattan Project was refining nuclear materials for both weapons and for energy. And because we are who we are, we tried our best to contain these materials when they were sent to waste. But as it turns out, we didn't do it very well, and it began to leak out. Now, what's kind of interesting is that one of these waste containment regions is this perched sort of pond here called the S3 ponds. There's actually 4 different units in there. And when we got sick of looking at it, we paved it over and we made it into a parking lot. And what's happened is that there's been a rupture. So there's water flow down Bear Valley. There's a little creek that moves down this valley, and water is flowing down this valley, moving materials up from the mountains down into the valley. And it passes across this pond, and this pond here, which contained the contaminants, has sort of broken. And sort of around here, there's a plume of nitrate and uranium and other metals and some organics. So the organics have largely been used up over the past 50 years. And it's moving down into Bear Creek and then moving down the valley. These are exceptionally high levels of nitrate and uranium. It's perhaps one of the most nitrate contaminated places on Earth. Not the most uranium contaminated locations, but it's very high in nitrate. And what I'm showing you here is these particular regions that this project has broken out to study. Now, this is emblematic of the type of information we have to capture in knowledge base about where things come from. And the sample work that Ben talked about is about representing where you're doing your work, what the nature of those areas are, and then how we sample from those locations. So we're sampling in a large number of locations along this site. As an example, we have just completed a study where in this Area 3 region, we have a number of groundwater wells for big holes we dig in the earth, where we stick instrumentation and we can measure the groundwater in there. And all these little dots you see are things that are called cone penetrometry cores. This is where you have a metal rod that we push into the earth at a constant rate. And we can measure based on sensors on the rod and in the truck itself, the nature of the sediment and the nature of the water as we're pushing through it. And as we push through, we collect the sediment and are able to, as a z-axis, get chemistry and biology in that sediment. We've built a very tight array of information along that side. And that allows us to build very sophisticated images of what is going on, for example, in that region. And so this is a three-dimensional cross-section where the colors are sediment types that we have, we've measured there. Blue, of course, is where we think that there's water and groundwater flow. And the little the sort of little poles you see in there, those are actual wells that we drilled, and we have constant observations going on that give us spatiotemporal information in this, informed by this region. So from the ecology standpoint, what this gives us is a view of the volume of Earth that these microbes are living in at fairly high resolution. Because we have a hydrogeology of the surrounding area, we kind of know how the water is flowing through this region and what it is bringing with it. And because we have sensors, in the sediment and in the water here, we kind of know a great deal about the chemistry and water flow. We have weather stations associated with this. We know what the recharge rates are from rain and other things. We have some idea of the temperature and wind and things like that. Now, when you look at one of these wells here, this is all space indexed. So we know exactly how tall the well is. It will be screened at some level here. I mean, there's water flowing in through some region of this idea. And that is a measurement of what we think the water is flowing in through this region here, and should be a measure of what microbes and other chemicals are present in that region. We've also scored the surrounding regions by the type of region it is. For example, is vadose, capillary fringe, or saturated region, for example. And when we do that, we can then measure as a function of z-axis and x and y, things like pH, or we have 53 metals that we measure, because we can measure that many metals. We have total organic carbon, total organic nitrogen, and we have a large number of metabolites as well that are present in these locations. Because it's cheap and fast, we have a lot of amplicon data. And this is just an example of amplicon data arrayed from one of our sediment cores. So this is a single time snapshot where he took a big bunch of sediment out of the earth, and we cleaved up that sediment into different regions by depth. And we measured who was there. And so on this axis, it's the sample, which is a particular XYZ location. And then on this axis, it's the exact sequence variants, or ASVs, where we can see that they're highly stratified in space in this location. And they have some amount of correlation or association with the chemistry that we see here. So again, this gets back to how we need to represent information in our system. We are trying to capture knowledge about where samples come from, what's being measured and how, the units of those measurements, of course, and all the protocols, and hopefully all the protocols that made them. And then we have things that go beyond amplicon and biogeochemistry and hydrology. We have things like metagenomics that gives us genes and mags and their distribution in depth. So it's a fair amount of information that we have here. There's more than that in our sample set, but this gives you some sense of the amount of data we're able to collect. And we're in the midst of building an observatory out there, which we were operating 24/7 for years, collecting this information in at least a daily sort of way. So this gives you some sense of how we're organized. We have our field group, which is out there building these observatories that have these wells with these observation ports in them. Allows us to measure both sediment and water in space and in time. We have some sense of where the water flow is, and so we know how these wells are connected. We measure the connectivity hydrologically and so on. And so then we're able to figure out sort of what the boundary conditions are for the community that we're operating. As we sample, we bring those materials back to the laboratory and we characterize them in great detail, but we also cultivate and isolate microbes. So what you're seeing here, for example, that this has not been built in KBase, this should be built ourselves, of all the taxa that we observe out there by 16S or by metagenomics. And the black dots are where we have isolates that we've been able to collect in the field that are exact matches at their 16S for the guys that we see out in the field. And then it turns out that we have a fair number of we prioritize these by their importance, that is how abundant they are, how active they are by things like bond cap, and whether or not they correlate with biogeochemical processes that we observe in the field. And so we need a way of turning this data into these trees, relate those to the individual isolates that we have, and then to score the isolates by how important they are for the biogeochemical processes we're observing in the field. That's sort of the workflow we're trying to put together here. And so this requires a fair number of data integration steps and analytical steps that link the data one to the other in reasonable ways. Once we have an idea of the most important microbes and how they operate, we have a characterization pipeline that measures their growth under hundreds of conditions. It measures their exometabolomics under a series of very well-chosen conditions. And it begins to, we sequence their genomes, and we can, in some cases, do genetics on those guys to figure out how each gene confers fitness in those environments. Once we have scored important microbes from the biogeochemical point of view and the ecological point of view out here, and we have their genome, their characterizations, we can prioritize those for creation of synthetic communities that are simulations of what we observe in the field, or are designed to prove a principle of what we observe in the field. Now, we're interested in nitrate reduction and the coupling nitrate reduction amongst community members. And so what we'll be showing you is this idea that we can pick these microbes out, assemble them, and begin to measure how they operate and predict how they'll operate using KBase functionalities. Once you figure out what the critical mechanisms are in the laboratory that we think are operating in these syncoms, we can then look for the signatures of those mechanisms back out in the metagenomic data. And this is just an example where we happen to have one of those cores I showed you, one of these cores that we take down one of these wells here, where as a function of depth, we expect to see two different groups of nitrate reduction genes to be differentially distributed, and indeed we see these differences in that distribution in space. Okay, that gives you some overview of what we're on about. So let's sort of think about how we drive these things, then we'll get into the KBase workflows. So we have this field observation that it started this all out, that as we were to look at pH, for example, in the field, we would see that there were certain regions in which nitrous gas, nitrous oxide, was released. Nitrous is a bellwether of incomplete denitrification. And so the thought is that pH is mediating when nitrate can be reduced in the field. And since nitrate is the thing we want to reduce, we want to get rid of it and turn it into nitrogen in this particular location. We want to understand what is causing the slowdown of denitrification and the release of nitrous gas, which is not good because it is a very potent greenhouse gas. We noted as well that the nitrous, that when rain occurred, oxygen would drop and pH would drop, which would then be correlated with the off-gassing of the nitrous itself. And so the hypothesis we came up with was that rainfall events at the FRC drive a drop in pH, causing microbial communities to shift towards nitrification. So we realized this, to prove this, we had to measure a number of things. Number one, the metals play an incredible role in this, because it turns out that at our site, things like molybdenum may or may not be bioavailable. Molybdenum is a metal center for denitrification. Number two, there may be chemodenitrification due to the iron oxides that exist at the site. And we want to make sure that we're accounting for the abiotic routes for denitrification. And third, that low pH was known to be a sensitive, known to be a controller for two of the most sensitive steps in denitrification in our system. So we wanted to make sure that we were measuring which of the enzymes were present and whether or not likely to be pH sensitive. Now, ultimately, our goal in Enigma is to integrate this information into a truly mechanistic model of what is going on inside the world. Now, this is a scary looking model. I'm not going to explain it all to you. We actually have published an explanation of this in this paper over here. But this is essentially a classical reactive transport model in which we not only measuring the reaction and transport of chemicals, but the transport of microbes and their activities, where we represent we represent their genetic capabilities for both growth and for interactions amongst the microbes and for chemical transformation, all in one model. We're right now doing the design of experiments so we can parameterize each of the mechanisms in this model along the way. Now, this is exceptionally difficult to coordinate all this data, to coordinate all the sub-analyses, to coordinate all the sub-models that can be built, including these genome scale models of the ability of cells to transform chemistries. And really, it's almost impossible to keep it together. When there's 20 of us or 30 of us working together to get this to happen on different machines in different locations. KBase is meant to solve that problem for us. And we've been working really hard with the KBase team, given that I have some privilege there, to help us get that job done. Luckily, a lot of the other community members face other jobs, and so they've added things to the system that make our job much easier. Now, Enigma, to do this, is a very large multidisciplinary group doing this together, and this is emblematic. One of the biggest complaints of new people joining the program was they couldn't find where our data and our analyses were. Now they don't worry about that. They can go to KBase. So let's talk about this. We're making lots and lots of data. So the first thing we do in KBase is bulk upload and organizing data with organism samples. Now, before I tell you about all this, I want to go back to what Ben told you. So Ben and his team and members of KBase in general have put together a fair number of tutorials, both written and video, that you guys should be aware of. This slide I'm showing you here is a subset of the tutorials that have been put online that are what we use to do the work that I'm going to show you next. What's really neat about this is it takes you through how just to organize your team and organize your data on system, how to take that data and turn it into the fundamental identification of the taxa and genetic functions that are present in your samples, and then how to turn that identification of taxonomy and genetic function into models of how the individual microbes operate and how they operate together as groups. So we have tools that take you from the very raw data all the way into models, metabolic models of the entire community. What's really game-changing, aside from being able to do this all in a single place, in a shared environment where you can collaborate on it, is that once you're done, it is easy to publish these results out into the world with a single click. You can get a DOI number, and you can get something that is available, and searchable by Google, and is a fully reusable, fair representation. It means findable, accessible, interoperable, reusable, representation of all the work you've done that is citable and otherwise. Once you have it, it can also be used by educators. And we, Ellen Dow and Alicia have done a spectacular team in building a fantastic team of people building educational materials based on these types of analyses we do here. As we move through, I'll show you what we're using along the way. So let's start with organizations in KBase. Ben mentioned this idea. My SFA has 87 members in our organization here, and we have 71 narratives. Narratives are the means by which we organize data and do analysis on that data in ways that are useful to people. And what's really cool is that we can see what each other are doing in real time and work together on those topics. An organization is a way of being able to present both to your own team, and when you're ready, creating ways of serving your groups of data and analyses to the outside world and getting credit for it. Now, of course, we had a lot of data, and it can be very painful to upload data into a system. And so the team has worked very hard over the past couple of years to give you the ability to upload large numbers of data, large numbers of data files in one fell swoop. Now, of course, because KBase is a knowledge base, we ask you to do things you might not be asked to do in other systems. We really do want you to say what your samples are, what your measurements are, and how they work. That does put a little bit of a bar on things, but the value you get back from that is extreme. You get back a lot of integration with existing information on the system that allow you to query in ways you can't query anywhere else. And we'll go through some of that later. Let's talk about bulk import. So bulk import automatic is a way of using of bringing many different files onto the system at once. Right now, you can do reads and assemblies, and you can do, I think, chemical abundance, more or less. So it can detect by file extension what kind of types you're looking at. You can import multiple files of different data types at once, and you can select the files to import through this GUI or by an import specification using a sort of a file that organizes all the file, that is sort of an index, a table of contents, if you will, for all the files you're going to upload. This made it much easier for people to get lots of data onto the system. And there's a you go to docs.kbase.us, you can find the bulk upload guide there. Now, as you load data into the system, you want to organize it in terms of samples. Now, conceptually, what a sample is, it's the physical element that you are observing in the field. And it's a particular set of measurements associated with a space-time sort of measurement of that sample. So in general, for example, if you take a piece of sediment from the field, then that sediment was taken from a time-space location in the field. It itself has some volume and some extent. And then we might make measurements of it, such as its pH, or all its chemistries, or its amplicon, content, or its metagenomic content, and so on. And we want all those different measurements to be associated with that single object, more or less. It could also be a location in a river where you're sampling out, water at a given time. But it's each one is a unique time-space sort of object that we're observing, and then a bunch of measurements associated with it. And then sample sets are sets of those. So that's sort of what I said here. You have this sort of sample. To do that, you have to have all the sample metadata. And that metadata would be geographic location, temperature, collection information, the environment, material, and so on. And one of the things we've really been working on is ensuring that these all adhere to ontological standards. That means that units are made formally, so you can cross-compare things of similar units, not comparing apples to oranges. That your environments, while you can describe them as you wish, you can also draw from, for example, the environtology. So you can say this is specifically, a terrestrial subsurface sediment environment, right? And things like depth and temperature are actual numbers that mean something. And geographic location is stored in a common, longitude, latitude, framework. So then you can link together these data. We have these sort of services to do that. And you can link these types of things together. All some things with metabolomics and chemical abundance, metagenomic sequences, and OTUs and amplicon data are right now what we support. What's neat then is that that makes it easy, because they're all in the set format, to bring them together and do group analyses in terms of like correlations amongst data, taxonomy of all the fellows in your, of all the microbes in your sample, their abundances, and doing comparative genomics. The other thing which is nice about this is because you can organize, experimental campaigns, for example, by the sample set that they're in, you can decide at the sample and sample set level who has access when to that data. Because even in the same project or same program, there's some sensitivity before letting your data out into the outside world, even to your collaborators, because you want to make sure that it's good data before they get it. So you have a system for sharing permissions and controls. For example, in Enigma, we have a series of campaigns. These are 4 campaigns that we ran. And so these samples are organized into sample sets. And these sample sets have lots of interesting information in them, including how many samples are in them, that's 128, how many attributes that we give to each sample, ours has a lot, 217, and what they have in them, amplicon, chemical, or paired end libraries, and so on. What's really neat is you can drill down on these guys. And so, for example, for that first one, so it's going, for that first one, you can get a sample from that set. It will tell us where it came from on a map, for example. It'll tell us what measurements we have each one of these locations. And this is where you can begin to do things like, figure out who has access. You can see that this sample has a lot of information about where it came from, about the sediment it was collected from, and specialized fields for Enigma, including, for example, how much nitrous there was being emitted at this location at this time. This is an example of the permission screen. So this gives you some sense of that I own these samples. I've assigned administrator access to some of my colleagues. These happen to be KBase people because they were helping us get this job done. And they needed admin access. I've given my KBase team read-only access because I want to have full control over these samples, so they don't muck up my particular organization. That said, they can take my samples and reorganize them to their own sample sets if they want, and copy my sample set and use it there. Okay, so now that I have my data on system and it's properly organized and I've shared it with the right people, we can begin to work together to do something interesting. So this sort of goes into how we use sample and amplicon analysis and samples and chemical abundance. So over the past little while, we've added a fair number of applications, we call them apps, on the system that allow you to create plots like you're seeing on the right over here. And these are things that do things like predict community functional potential. Examples of that include FAPROTAX, which is a way of going from taxonomy to projected traits, microtrait and DRAM that use curated hidden Markov models, more or less, to assess whether or not your genome or metagenome has a certain genetic potential. And then you have tools for doing both alpha and beta diversity calculations, and then to correlate taxa with their environments. That is, which taxa or which are correlated with, for example, nitrous production or pH or so on. So this allows us to do workflows like this. This is just an example workflow. I should say that in KBase we don't really have canned workflows. We do have narratives that you can use, and you can click through what someone else has done, but they're not really sort of workflows. You get to choose along the way what you're doing, and choose the parameters you want to use and choose the order in which you do things. So in this particular case, we at the moment don't allow you to do 16S read analysis on the system, but you can do that read analysis off system and create amplicon OTU tables and 16S sequences. You can upload an amplicon matrix. You can then transform that matrix to normalize it in various ways. You can then do the same thing for chemical abundance and bring that onto the system. In this case, we're talking about a particular campaign, the 100 well survey that we did. Then we can, because they're from the same set of samples, we can then cross-correlate, for example, taxonomic abundance with chemical abundance and get these sort of matrix of chemical health correlations. That's an example of getting this sort of analysis. This is a segment from KBase through that table. You can do this on KBase. And here you can see, for example, that there are ASV that have very strong positive correlations with nitrate and nitrous and pH, for example, simultaneously. Now, the Enigma researchers who are looking at this could take a look at this table and look at these ASVs. They could say, you know something, the number of these ASVs here, for a number of interesting reasons, it turns out that for both nitrous and nitrate, this OBA, blah, blah, blah guy seems very important. We happen to know that that's kind of interesting guy because it turns up in a lot of our analyses. And so we select these two guys out as interesting fellas. So now we can think, well, who is that guy? What is it doing? So we can run this through things like classification tools. And so we can import the amplicon matrix. With that comes the V4 regions for the 16S. In this particular case, we then do an RNA classify with RDP. There's other ways. I think there's other classifiers in the element system as well. And this will give you, for our amplicon matrix, a set of taxonomic abundances along with it. This is a sunburst chart for that. And down here is that OBDA. You can see it down here. It will end up being the OBADA amplicon. And what we are able to see when we look at that particular data, you can sort the samples by nitrous oxide concentration, for example, and then plot the abundance of that particular ASV, on that axis, and you can sort of see that it really is enriched at very high nitrous levels. Now, why might that be? We can take a look more at the metagenomics, for example. This, by the way, that was Rodanibacter that we came up with. It ends up being classified as Rodanibacter. Rodanibacter is a known denitrifier. So now that we have metagenomic data that complement the amplicon data, we can bring all the other tools into play. And these include detection of novel lineages, detection of viruses, detection of the functional, we can create MAGs and do functional taxonomic profiling of MAGs, which are metagenome assembled genomes, and we can do comparative analysis of it. So these are examples of looking at data from one of our sample sets, which is the ED106 high nitrate core. So this is the S3 pond I told you about, where all the waste was. We took a core sample right below where the plume comes out, which is right over here where this red circle is. And this is the interface for samples. We clicked on that particular sample set and asked where it was, and it's here. These are all the samples in that sample set. These samples are represented by lines on this z-axis for the core. So each one of these samples here represents one of these lines. And the color of it is what zone it's in. And so we can take a look at, for example, moisture as a function of depth, pH as a function of depth, and measure as a function of depth. So you can see there's lots of different combinations of parameters here. And these combinations are what we think may be selecting which organisms are present. So we can then ask from the metagenomic data, let's match this chemical and physical data with taxonomic data. So with the same sort of depth, we can do it by here. And you can see there's sort of these differential groups of taxa at each one of these levels. And you can see that in this region here, corresponds to this region here, you end up with this sort of blooming. This region here, so this region here, this region here is this region here. This one maps to there, this one maps to there. That there's a sort of blooming of these couple of clades that come out. Similarly down over here. There's a couple of clades that come out. So those end up being very interesting. And when we look at those, there's a lot of Rhodanobacter and a lot of these Burkholderia sort of borax type guys. And a lot of Rhodanobacter. So because the OBABA mapped to Rhodanobacters, and because we see the Rhodanobacters expanding in those regions, we can then take a look at what genes are also present. And again, we can look at these sort of regions, 1, 2, 3 regions, where there's a large abundance of specific nitrate reduction genes that are present, for example. So that's a hypothesis that those are being selected there because there's high nitrate through the plume coming through that region of that three-dimensional setting that I showed you earlier. And when we look at the MAGs themselves, all the MAGs that we assemble, and this is the workflow we use for creating the MAGs and the metagenomic data on KBase, and then we annotate those MAGs using microtrait, which came from Ulas Carrallas and Owen Brody, also on our system. We're able then to map so there's nitrate pathway genes, not just from the whole metagenomic assembly, but to those individual MAGs. So we can identify specific MAGs that were important. So now we have the MAGs here which we have a lot of genomes in the region of interest of these two types, the Burkholderia and the Xanthomodalis, which are where the Rhodanobacters are, and some pseudomonads. And then we can go to the laboratory. Now, during the same time we're collecting that sediment, we were actually enriching microbes from that sediment and from that sediment, other sediment. And we found a lot of Rhodanobacter, pseudomonads, some Rhodanobacters, and a lot of Burkholderia from the cultivations. So the cultivations are a bit biased. They like weedy, fast-growing organisms. Rhodanobacter's not so bad at that, but we've only got a few Rhodanobacters out of that guy. But now we want to see if any of these guys map to the critical MAGs that were sitting out and in the field. So when we take a tree of all of these guys, take all the MAGs and isolate genomes, and we build trees of them in KBase, and there's a narrative that was made by Dylan Shidian that goes through there. And you can follow along with that narrative. You can find out that in this tree, you can see that there are these organisms have different amounts of nitrate reduction pathway in them, and that the Rhodanobacters come in a couple of flavors, guys who have it all and guys who are partly present. These are all the high quality MAGs, and it turns out that the isolates aren't quite the same guys as the MAGs. They're not quite lined up, unfortunately. They can be close, but they're in a separate clade. You can kind of see that. That's unfortunate, but you do it. Now we know we have to go back and really work on trying to find those Rhodonobacters. That's useful for Enigma. But we can begin to also think about studying the guys who are closest in to the Rhodonobacters that are closest to the ones we see in the field. We also have the Burkholderia. These are a much larger set. These are much weedier organisms. So we have many, many, many more of them. The ones that map to MAGs are out over here. And here, the MAGs and the isolates are mixed. So we have guys who are very, very close to the acidophorexes in the field. So we can blow that up into a tree. This tree wasn't built in KBase, but it sort of summarizes a bunch of things for us. We're actually trying to get a tree like this built in KBase now. This is actually Y12. But what's nice about this tree is you can see the organisms, both the Rhodonobacters and the acidophorexes we're interested in, and other things, treed up. And you can see where there are isolates, these blue circles, and MAGs in the white circles, and see where we can find guys who are likely to have linked to an ASV, have an interesting nitrate reduction pathway. In this case, this one has incomplete nitrate reduction, and this guy has complete nitrate reduction, and where we can get guys who are very close in isolate space to both these guys. So those are two guys. And so, more or less, we have a hypothesis that these two guys are found together in the field. They occur in places where when pH changes, the nitrous gas changes, and that we hypothesize that this can occur when there is a handoff of materials between complete and incomplete denitrification of these two strains, and where the enzymes that execute these things are differentially sensitive to pH. So now that we have these two guys, we have the two isolates. We also use KBase to assemble them, to annotate them, and to make ultimately metabolic models of them. So if you want to know how to do that, there's an introduction to genome analysis here. And it turns out that when we were doing this, we came up with a bunch of cool isolates. One ended up being very cool because it was very metal tolerant, among other things. It wasn't either Rhodonobacter or acidophorax, but it was so cool. One of our group members followed it up, and they just published a genome of it recently. I'm bringing this to your attention for the following reason. In the course of research, you often get these things that come up along the way that you can publish, and you wanted to do that. This is a common waypoint in mechanistic analyses. You find a new genome, you assemble it, you annotate it, you annotate it very deeply, and KBase allows you to build these sort of genome announcement papers that have a lot of depth. This is an example of that, where Jennifer Goff, who's at the Michael Adams lab at Georgia Tech, who's part of Enigma, made a static narrative where she did all this beautiful analysis, including genome assembly and quality assessment. She annotated them, she did classification with taxonomy, she did a metabolic and put it into taxonomic context with other members of this clade, back to the serious strain. Did some pangenomics on it, and also did metabolic modeling and flux balance analysis to show what its specialized capabilities were. She then created a static narrative, because once this narrative was built and she liked it, this thing you're seeing here is actually a published narrative. You hit a button and it ends up outside the KBase password level, and it allows people to see what you did, search for it on Google, and cite it. And if they want to, they can go into KBase and completely re-execute your commands, reuse your data, change parameters, and do what they want. And this is a paper that was just published. It was in Environmental Microbiology, and it cites this paper, this thing in there by its DOI number. So it's very, very cool. A perfect adjunct to these publications, and allows fast reuse of your information and propagation of this data on our system in KBase. So that last piece was doing metabolic modeling. That was one of the things, special things we do. Chris Henry is one of the world's experts in metabolic modeling, and his team has done spectacular work in this area. And he's made that very available to everybody on the system. And it's kind of a superpower that we have. One of the things we were doing in Enigma was taking these strains and growing them together. This gives you some examples of the things we're trying to explain. This is the data we're trying to explain. So we have them growing in denitrifying conditions, and this black curve is a pseudovorax, and it grows and it sort of peters out here, the low carrying capacity. The orange is Rhodonobacter growing alone, and it kind of keeps on growing. When we grow them together, the whole culture grows faster and gets the higher carrying capacity. If you look at nitrate, nitrite, and not nitrous oxide in the single cases, So in the orange alone, nitrate gets degraded at some rate. Same thing in the acidovorax alone, it gets degraded at some rate. There's a switch that occurs here. And together it's degraded much, much faster. Nitrite is seen both in the individual culture of Organibacter, but not in in the individual culture of acidovorax. Sorry, I did that wrong. It's seen in the individual culture of acidovorax, but not in Organibacter. And then together you can see it's been minimized. Nitrous oxide is only off gas in the individual cultures and not when they're together. So there's this idea that maybe they're collaborating to get the job done. So you use these beautiful tools that Chris Henry and his team have built. This includes ways of visualizing data on pathways. There's amazing extra pathway tools. Extra tools come from the Paulson group, I think. Fitting the model to metabolomic data that we have. We didn't use pickaxe, but those of you who are interested, this is a very cool way of making your models a lot better by figuring out what's nearby in chemical space when you don't have an annotation for a gene. So we use this workflow in KBase to annotate in a common way our genomes, to build metabolic models, to refine those models by fitting them to the exometabolomic data that we developed for them, so the models were more predictive of how they grew in the real world and what they made and ate. We then use the modeling, we then merge them into a common set of models, and then we ended up fitting that model to the exometabolomic data of the community. And now we had 3 models: Organibacter alone, acidovorax alone, and the community together. And now we can run these experiments, the experiments that I showed you back here, but on system using these fit models. One of the cool tools you can do is you can use this thing called a phase plane. And what it is you run the system under some varying parameter. In this case, it's how much nitrate is available for uptake in the environment. And you can take a look at, for example, how the community grows as you add nitrate, which is a receptor. And so what you find out is that at low levels, as you increase nitrate, you get better and better growth in this community by our model. That this line is equivalent to this axis more or less. You see this increase in acid uptake rate, and then it sort of peters out up here and we saturate. We can still grow even more as we increase nitrate because of other things that are being taken and used. And then finally, we can look to see where the denitrification is occurring. And so in this particular case, we find that at low levels of nitrate, it's the Organibacter that's doing all the work, making N2 from Organibacter. And at very high levels of nitrate uptake, it's the acidovorax who's mostly making it until it begins to peter off because of other reasons. And in the middle, there's the switchover point, the switch point. And so we use the modeling to try to figure out where that occurred and why. So this is an Escher map that kind of shows you what's going on. Initially, sort of at this lower region down here, the nitrate is entering both cells, but this is much more efficient at processing this through this entire pathway by the model. And so it turns out that Organibacter could gain all its energy by pushing the nitrate through the entire pathway and then competing for that substrate against acidovorax to create nitrogen over here. The acidovorax doesn't have a complete denitrification pathway, it turns out. It is predicted to make ammonia, for example. So that was the prediction was that initially everything was going through this route here. Then as you begin to get to higher and higher nitrate levels, the inefficiency of using these downstream enzymes to provide energy for Organibacter, when it begins to not need them to grow, leads to a overabundance of nitric here, and then that nitric oxide here, and that's able to be exchanged across the cellular membranes, and it can drive the downstream part of the pathway that the acidovorax has, even though it doesn't have a connector from NO2 to NO. So now you can, at this minimum region, begin to pass through this path here, and that is what happens during that switching thing. And then overall, the culture is able to grow because now we're driving energy for both organisms using the overflow metabolism of the first. Now, when we showed this to the Enigma team, they're like, that's really interesting, but we know some things you don't. Even though this Rodanobacter has NOS-Z, we have no evidence that it is active at all. And we've been able to knock it out and nothing happens. What's more is we have no evidence that this is confirming any ammonia. And so we don't think this is happening either. And so when we ran the models with those constraints, saying this doesn't happen, we instead found that as we move into the high nitrate regime, we begin to take in nitrate in both organisms. There's an exchange from acidovorax to Rodanobacter here to get it to NO. And then NO is converted to N2O, and it's the N2O that is transferred over to the acidovorax and turned into nitrogen. And this is the actual path we predict going on. And now they're going in and trying to figure out if that's really what's happening. But it demonstrates this sort of cycle where the models help us build interpretations. Protein organisms are working together to process nitrate differently as its availability changes. So to summarize what we did there, this workflow identified from the field data the top species correlated with critical field process parameters in genomes and genes stratified along those gradients. So we knew which systems and which organisms were likely to be important. This set priorities for the isolates that were likely to have properties of interest to the field that could be analyzed in a laboratory. We were able to analyze those by assembling their genomes, annotating them, and understanding their functional capabilities. We were able to then use that genomic information in comparison to MAGs and other things to figure out which two to bring together into a syncom, to explore how cross-coupling denitrification could occur in various scenarios, for example, in increasing nitrate. We then showed that you could shift community productivity and which microbes are likely to be the important ones as you shifted across the nitrate gradient. We were able to show their initial guesses for how that worked in KBase were wrong, but then revise the model along the way to suggest how they actually operated. And now we can collect the field and look for signatures of these. And if this proves correct, this narrative that we've created and then validated is shareable with the world immediately and is propagatable to everyone on the system and who searches for nitrate reduction and Rodanobacter or acidovorax, which end up being important across nitrate contaminated sites. So this really is towards a mechanistic ecology workbench. It allows us to organize complex multi-omic samples, link amplicon abundance to numerical measurements, combine these with the assembly and annotation of metagenomes and the mapping of traits, and mapping to their traits and their functions, and integrate that with laboratory data on isolates to really come up with a fundamental mechanistic model of what we think is happening in the field. It's about 25 apps, around 10 data types, and now instantly accessible to everyone on our team. And we publish it using static narratives, instantly accessible to all of you. And I think that is immensely powerful. You can check our work, you can have our data, you can change the parameters, you can call us into question, and that's what science is about. So now that I've told you that, I can tell you what's next for KBase. We're, of course, expanding our apps and data types that we're supporting. We were working with people like GenPet Ridge on stable isotope probing. We're working on new ways of incorporating activity data and genetic data. It's going to be very, very cool. We're coordinating how samples are labeled and annotated across different resources, such as the Drug Genome Institute, the National Microbial Data Collaborative, and ESS-DI. And that's really powerful because it means if you are working with one of those agencies and you get data into their systems, you'll be able to transparently move your data from one place to the other, maintaining their sample information. So enter once, use everywhere. We're enhancing the publishing of static narratives so they're richer. And what's really exciting to me, and Shane and Paramveer are leading this, is providing this richer knowledge environment so we can place your data in the context of all of the data on the system. So when we were doing this analysis, one of the things that was very powerful was being able to show where the microbes that we consider to be important for nitrate reduction are also found in other metagenomes across the planet, and whether or not those were nitrate contaminated locations. So it speaks to the generalizability of the models that we're making in the system by instantly integrating it with everything else that can occur in the world. And so the next few years, we're really drilling down on how to make this sort of search engine and this predictive analytical engine really work for our users and make these sorts of predictions. So from taxa and genes to the the bio that they live in, what processes are likely to be significant, and to predict what functions need to be present that haven't been observed already. So really our job is to continue to improve this mechanistic ecology and synthetic community engineering. Okay, with that, I will stop. Thank the team who really did this analysis. It was really, the KBase team really stood up and worked very hard to make all this come together and make sure the system worked this way. And they did an amazing job. And then with there, I'll also stop. And if there's any time left, or any time to take questions, I'll take them.
All right, thanks so much, Adam. That was a fantastic presentation. There was one question in the Q&A that I tried to answer there, but I'll just maybe you can verbally answer it. Is do you think that the phylogenetic difference between the isolates and the mags in your samples was due to the mag assemblies, or are there different strains that are yet to be isolated in that sample?
Well, no, I actually do think they're different. First of all, there's a bit of evidence even from the isolates that we have, that the Rodanobacters, even who are closely related, even however have small genetic differences, have very different sensitivities to the environment. And number two, the mags in our group are extremely high quality. They were produced both by short read and long read sequencing and assembled by Torben Nielsen and Lauren Louie, who actually put their long read their mag assemblers onto KBase and are now working for the long read assemblers onto KBase. And I have very high trust in those genomes at the moment. And the genomes we have in the laboratory are unfortunately relatively distant, even though they're the same genus, and in some cases the same species of what we think are out in the field. Yeah.
Looks like some folks are typing a question into the Q&A right now. It says, could you detect ammonia oxidizing archaea in your soil samples?
Oh, really good question. Someone out there is an ecologist. There's evidence in the ASV data that they are there at low levels. David Stahl and his laboratory are specifically working to isolate them, and we have some initial results that are promising in that direction. So we do think they play a role, and we do think they're important. Yes.
All right, I don't see any more questions in the Q&A doc. Maybe if anyone has a verbal question, they can use the raise hand function on Zoom.
There was a lot to take in.
All right, seeing no more questions. Yeah, thanks again, Adam, for this fantastic webinar. It was great to see all of the integration of KBase features and the data that you use to do this analysis, and you get a little more depth of what you did compared to our speed run for GSP. So thanks everybody for attending this webinar today. We'll post this up on our YouTube shortly if you want to review it later or share it with your colleagues. And yeah, thanks everybody for attending, and we hope to see you on one of our upcoming webinars soon.
Thanks, guys.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.